Hello, you are welcome to Integrated Science Intervention Program for Senior High Schools. The topic for today is Variation and Inheritance, and the lesson title is Monohybrid Inheritance. I am your facilitator, Gilbert Odey Into. Now for a starter, let's look at these two plants on your screen. Let's focus on the color of their flowers. You can see that one has red flowers and the other has white. Now, what do you think will be the flower color of the offsprings if a cross-pollination occurs between the plant with red flower and the one with white flower? You have just 10 seconds to think about that. Yes, you may have thought of so many answers, but let's come together and summarize them. They may produce young ones with red flowers, which will be dominant for the red flowered plant, or ones with white flowers, which will be dominant to the white flowered plant, or a mixture of white and red, which will produce pink flowers. Today, our lessons will be on how we inherit such characters. For our lesson outcome, we will learn to define the term monohybrid inheritance and give examples. We will also state and explain Mendel's first law of inheritance. We are also going to learn about how to make monohybrid cross diagram using Mendel's first law of inheritance, and finally we will state some characteristics of albinos. For our outline, we'll look at monohybrid inheritance, we'll look at Mendel's experiment and the first law of inheritance, and we'll also look at albinism. Now, the words that are going to feature in this lesson are monohybrid inheritance, monohybrid cross, Mendel, albinism, and segregation. Now let's move on. What do you understand by the term monohybrid inheritance? Monohybrid inheritance refers to the inheritance of characteristics which is controlled by a single gene. By that, we mean the transfer of characters from parents to their young ones. And in this transfer, the characters that are transferred are controlled by just a single gene instead of several genes as we have in some cases. For example, we have albinism, which is controlled by just a single gene that is responsible for the non-production of skin pigment. We also have the color of flowers like we dealt with initially, which is also controlled by just a single gene. And also have the eye color of humans, which is also controlled by a single gene. Now let's move on. This monohybrid inheritance idea was brought up by a man called Gregor Mendel. This man is an Australian monk. Though he wasn't a scientist, he was interested in gardening. And in his garden, he planted pea plants. I hope you can see the pea plant on your screen. The pea plant is what in Africa or in Ghana we call beans. He observed them during the planting seasons And through his observation, he saw so many different characters. In one of his observations, he chose the heights of the plants. He cross-pollinated a pure-breed tall plant with a pure-breed short plant. By pure-breed, we mean the tall plant is really tall with no shortness gene in it, hence cannot produce any short plant. Likewise, the short one. The short one too, pure-breed, means will not produce any gene that will lead to production of offsprings who are tall. After cross-pollinating these two, the seeds he got, he replanted them. And to his surprise, they produced all tall plants. So he was thinking, where has the shortness gone to? Because he crossed two plants, one short, one tall, but all their offsprings were all tall. So again, he recrossed the offsprings who are tall by self-pollinating them. So he pollinated their flowers, and the seeds they produced, he replanted them. Again, to his surprise, he saw that they produced now tall plants and short plants. But anytime he self-pollinated them, the young ones they produced had more tall plants than short plants. After that, he came out with a law which is known as Mendel's first law of inheritance, or the law of segregation of genes. This law states that the characters of organisms are controlled by a pair of internal factors called alleles. And these alleles separate in gametes. Per this, he means that all organisms' characters are controlled by two genes that occupy the same relative positions on similar chromosomes, of which one comes from the mother and the other comes from the father. And during reproduction, they separate in the sex cells. That is, the sperm will have only one of such genes, and the egg will also have only one of such genes, so that when they come together again, the pair will be formed. Now, let's see how this is represented on genetic cross diagrams. In your exams, you will be given such a scenario and be asked to present them on genetic cross diagrams. Let's first see the first part of his experiment. In the first part, he crossed pure-breed tall plant with a pure-breed short plant, and all their young ones were all tall. Per the genetic cross diagram, we first bring the parental phenotype, which is the physical characters of the parents, and in this case is a tall plant and a short plant crossing. So the cross is represented by a times sign or a cross sign in between them. The next is parental genotype. Per genotype, we know that they are the genes that control those characters we spoke about. In the case of the tall parent, we got to know that it is pure-breed tall. That means it is homozygous. And that means the genes that controls that character are similar. Let's look at something before we represent that. Normally, we choose a letter for this character. And if the letter is not given to you in the exams, you choose the first letter of the dominant character. In this case, it is tallness, because per his experiment, tall plants keep on occurring at higher ratios. So here we choose a capital T, and we double them because Per the law, all physical characters are controlled by internal factors which are paired. Then the small letter of the capital letter we've chosen will represent recessive because they are alleles. And we know alleles are same genes, but they control contrasting characters. Again, we bring our times because the times shows that they are crossing. Now the next is gametes. We said in the gametes, the alleles separate or the genes separate. So here we separate the genotypes. So we write just a single T and a single T for the tall plant, then a single small t and a single small t for the short plant. But please note, here we circle them. The circles show that they are gametes and not genes. Now let's come to fertilization, which is the cross. First, the first capital T will take the small t of the second one, and they will produce capital T, small t in the zygote. Again, the same capital T will take the second small t and it will produce capital T, small t. Then we take the second capital T, which takes the second small t, also to produce capital T, small t. And finally, it takes the second capital T takes the first small t to produce capital T, small t. Over here, these ones are called F1 genotype. And F1 means direct offsprings of parents. Now let's see their phenotypes. Because they all have capital T's in them, they will all be dominant. So they will be tall physically. And this will represent the F1 phenotype. Now for the second part of the experiment, he recrossed the tall plants that were produced in the F1 generation, and he had tall plant and short plants at their young ones in the ratio 3:1. Now let's see how that is represented. So again, we have the parental phenotypes crossing. Then now the parental genotypes change. It is now capital T and small t because it is from the F1 generation. Then for the gametes, again, they separate like we did for the first one, and we circle them, showing that they are gametes and not genes. Now we come to the crosses. In the crosses, the first capital T takes the second capital T, and we have capital T, capital T. Then again, it takes the small t in the second parent, and we have capital T, small t. Then we come to the small t in the first parent, taking the capital T in the second parent. But here we don't write small t first, we write capital T before small t because the capital dominates the small letter. Then finally, the small t in the first parent takes the small t in the second parent, giving small t, which is two recessives. Now this is F2 generation because they are young ones of F1 generation. Now let's come to their phenotype. Phenotypically, the first one has capital T, which is double capital T, and hence it will be tall. The second and the third also have capital T, though small t is there. The capital T is dominant, so they will also be tall. But in the last one, it is two small t's, and hence that particular plant will be short. So per his experiment, he got F2 generational offsprings to be tall and short in the ratio 3:1. And that is what is governing monohybrid inheritance up to date. Now let's take an example of monohybrid inheritance, which is albinism. You know much about albinism, but now let's dwell into it and get a lot of information about it. Albinism is not a disease, like many people think. It's not a disease. It's just a genetic disorder that is characterized by the non-production of skin pigment melanin. So such persons are not sick, just that they cannot produce the skin pigment melanin, which gives the skin its normal color. This is controlled by a recessive gene, which is inherited from both parents. That is why we say it is a genetic disorder. And genetic disorders cannot be corrected because we inherit it from parent and it is on our gene. Now let's see this family on your screen. We have two parents who have dark skin giving birth to a child who is an albino. This shows that it is recessive in both parents. That is the reason why the child got the two recessive genes and became albino phenotypically or physically. Now let us use a small a as recessive gene for albinism and capital A as a dominant gene for normal skin pigment formation. Let's see the ratio in human population for genotype and phenotypes. If a person has genotype capital A, that person has normal skin pigment and hence produces the normal skin color. The next one is capital A, small a. Such persons, though have small a, the capital A dominates the small a, and hence such persons will not be albinos. They will have the normal skin pigment, but will be carriers of the albinism trait. So such persons are known as carriers of albinism traits. Then finally, we have small a, small a. Here, they have both recessive genes. Hence, they have no option than to be albinos who cannot produce the skin pigments. Now, let's see how this is represented genetically. The cross between two carrier parents, like we saw in the picture, will be represented like this by genetic cross diagram, which we need to know for our exams. First, we come to the parental phenotype, which is carrier of albinism crossing with carrier of albinism. For the genotype, they both have capital A, small a, because they are both carriers. For the gametes, they separate and they are circled. So one of the gametes will have capital A and the other will have small a for both parents. Now let's come to the crosses, which will be the possible genotype of the young ones. First, we have capital A taking capital A and we having genotype capital A, capital A. Second, the capital A takes small a in the second parent and we'll have capital A, small a. Likewise, the small a taking the capital A again in the second parent. Then finally, if the two small a's meet, then the genotype will be small a, small a. Now we come to the phenotype of the young ones. The young ones that will have a pure breed normal skin person who produce the normal skin pigment. Then we have heterozygous offsprings. That is the ones who are carriers of albinism, two of them. Though they will be normal skin, they will have albinism hiding in them. Then unfortunately, the last one, which is small a will be an albino. So that is the reason why two normal skin parents can produce albinos. Try and cross for other parents. For example, you can try your hands on a normal skin parent with no albinism, marrying an albino, and see what will happen. For two albinos, when they marry, definitely they will produce albinos. Now, let's come to characteristics of albinos. If a person inherits the gene of albinism, that person will definitely have a very light skin color because of no skin pigment melanin. That person also has hair color that ranges from white, yellow to brown. That means that person may have white hair or yellow hair or brown hair like you see on your screen. Again, the eye color ranges from brown to pink. For brown eyes, we all know how brown eyes looks like. It looks like ours. But for the red eye, look on your screen. You see the person's eye is red or pink in color. And that is what some albinos suffer because of lack of the pigment of the eye. Then they also have poor vision because of the lack of pigment on their retina, which is sensitive to light and gives good vision. They also appear slit-eyed. That is, their eyes are almost closed due to sensitivity to bright light. So if you meet an albino in a bright sunny day, you think that albino has closed their eyes. It's not so. It is because their eyes are very sensitive to light due to lack of pigment. Now we come to the end of our lesson, and for our summary, we've looked at monohybrid inheritance and their examples, Mendel's first law of inheritance, and experiment using pea plants. We've also looked at monohybrid cross diagram of Mendel's experiment, and finally albinism in humans. You may try your hands on this question. A man who is an albino married a woman who is homozygous normal skin. That is, the man is albino, but the woman doesn't have any albinism trait in the body. Use genetic cross diagram to show which ratio of their children will be albinos. Use small a to represent albinism and capital A to represent the normal skin color. Thank you and bye-bye.